Thank you everyone for joining us and welcome to the Cancer Research Institute's webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Tuesday, March 18th, 2014. The topic for today's webinar is Immunotherapy and Colorectal Cancer. My name is Brian Brewer, and I'm Director of Marketing and Communications for the Cancer Research Institute. CRI is a nonprofit organization established in 1953 with the goal of advancing treatments that strengthen the immune system's power to conquer cancer. We fund scientists around the world whose work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 45 minutes, you'll have an opportunity to hear firsthand from an immunotherapy expert how cancer treatment today is undergoing a revolution thanks to these breakthroughs. After a 25-minute presentation from our speaker, we will open the discussion to questions submitted by you. You can pose your questions at any time throughout the presentation by typing in the Q&A box on your screen. Today's webinar is being recorded and will be made available for viewing on the Cancer Research Institute website and on our YouTube channel. Now it's my pleasure to welcome today's speaker. Dr. Lawrence Fong is an Associate Professor of Medicine in the Division of Hematology and Oncology at the University of California, San Francisco. His work focuses on developing immune-based therapies for cancers, including prostate, kidney, melanoma, and colorectal cancers, and he's contributed to research on 2 recently FDA-approved cancer immunotherapies. Dr. Fong obtained his MT at Stanford, completed internal medicine training at the University of Washington, and oncology fellowship at Stanford. He currently serves on multiple committees at the U.S. National Institutes of Health, and he has served on the editorial board of the Journal of Clinical Oncology and currently serves as a senior editor for cancer immunology research. Dr. Fong is pioneering powerful new approaches to treat cancer with the immune system, and it's an honor that he could join us today. As a reminder, we encourage you to type your questions into the Q&A box. And with that, I'd like to turn the presentation over to Dr. Fong. Dr. Fong, if you will, please.
All right, thank you, Brian. So I'm going to talk about immunotherapy of colorectal cancer. As Brian mentioned, we actually have interest across many different diseases, but colorectal cancer is what we'll focus on today. The outline of my talk is I'm first going to just briefly talk about colorectal cancer, just because I understand we've got quite a diverse audience. And then I'm going to go through our current treatments for colorectal cancer, and then talk about developing immunotherapies for colorectal cancer, and then finally finish with the challenges that we face in the field. So just a point about colorectal cancer, it's actually the 3rd most common cancer in terms of new cases in both men and women, and it also is the 3rd leading cause of cancer death in both men and women. And so it's an incredibly common disease, as I'm sure all of us are aware. And another point that I'd like to make is with regards to colorectal cancer. You'll see in my slides that I've abbreviated that CRC, just so you're aware of that abbreviation. But an important aspect is that actually the majority of colorectal cancers actually, unfortunately, identified once it's either spread regionally or actually with distant metastasis. And so this is really an enormous problem in terms of treating this disease, just because many of our treatments in these disease settings are unfortunately at the present time not curative. Now, when we think about colorectal cancer, we think about how far the cancer has actually spread. And so this is just a cross-section of the colonic mucosa, or the lining of the colon. And the main point is that as the cancer, which starts out on the surface of the colon, invades into deeper structures, it basically has a higher, what we call T stage. And once it's spread to lymph nodes, then that puts it in a different category, as denoted here. And then once it's metastasized to other tissues, it becomes metastatic. The main point here is when we think about these advanced forms of colorectal cancer, unfortunately, with regards to a 5-year survival with surgery alone, you can see that half of the patients with disease spreading to the lymph nodes will be alive at 5 years. And in fact, if a patient has metastatic disease, unfortunately, their expected survival at 5 years would be less than 10%. Now, when we think about how colorectal cancer can spread, lymphatic invasion is what we often see. And obviously, being interested in the immune system, this is an important aspect with regards to this disease spreading through the lymphatics. It can also extend directly. So these tumors, as I mentioned, typically arise on the surface of the colon, but then they can directly extend through the colon and into adjacent tissues. It can also cause a spread through the peritoneal surface, the lining of the intestines on the outside, and cause seeding through that route. And then the other aspect is spreading through the blood. So cancer cells making their way into the blood and spreading to other tissues. Colorectal cancer, by and large, when it spreads this way, impacts the liver first, followed by the lung, and then the bone and brain are involved less frequently. Now, in thinking about treatments for metastatic colorectal cancer, the the mainstay of treatment is a chemotherapy drug called 5-FU, which, as you can see, has been around for quite some time. And over the last decade or so, there's really been a huge push with novel chemotherapies, irinotecan, capecitabine, oxaliplatin, which are used in combination. And then most recently, has been the development of antibodies that have been used for the treatment of colorectal cancer. And I should point out that the use of antibodies are in many ways actually using, in this case, an immune molecule to actually target cancer. And we'll just touch a little bit more about that just to drive that point home. One of these approved drugs is Bevacizumab, also known as Avastin. And what that is an antibody that basically binds a protein that's produced by tumor cells called VEGF. The main point about VEGF is this is a growth factor that drives the development of small blood vessels, or what we call angiogenesis, that help to feed the tumor. And it's been borne out that if you use an antibody to bind up this protein, it basically prevents it from binding its receptors and generating those new blood vessels. And so this is one of the ways where immune molecules can be used actually in the treatment of colorectal cancer. And in fact, if you look at how this was shown to be effective, this is IFL, this abbreviation is basically for a combination of chemotherapies that I won't get into. But the main point here was in a large randomized trial that was published in the New England Journal 10 years ago, basically showed that if you added Bevacizumab on top of that, this antibody that basically binds up VEGF, you basically could improve the time to progression as well as the overall survival for these patients with metastatic colorectal cancer. Now, when we think about other antibody treatments for colorectal cancer, we talked about Bevacizumab or Avastin, but there are also antibodies that target molecules on the surface of colorectal cancer cells themselves. And in this case, we have these two drugs, cetuximab and panitumumab, which basically target EGFR, which is a protein that's also expressed on colorectal cancer cells. And the thinking here is that this antibody could bind these receptors on colorectal cancer cells and actually block signaling that may be driving these cancer cells to grow. But also, as it turns out, these antibodies can also serve to recruit members of the immune system, just because our immune systems actually recognize cells that are coated with antibodies, and cells within our immune system, including natural killer cells, or what's designated as NK cells, as well as macrophages, have receptors, or what are called FC receptors, that basically can recognize cells coated with antibodies and can actually go forth and kill these tumor cells. And so again, these two drugs are currently FDA approved for the treatment of colorectal cancer as well. Now, an important part of this is that you can use these antibodies alone or in combination with chemotherapy. And what's been important for the field is studies have looked at whether or not the tumors express EGFR or do not. This has not turned out to be a great biomarker or a great way to determine whether or not a specific patient's tumor might respond to the treatment. And I think this is one of the challenges of the field, is figuring out how we can better identify who's going to respond to different therapies, and that also includes immunotherapies. Now, to move to the next portion of the talk, what I'd now like to do is actually focus on developing immunotherapies where research has shown activity of the immune system in colorectal cancer and how we can actually develop that into immunotherapies for this disease. Now, when we think about how the immune system could potentially see a tumor, what we think is going on is that a cancer cell, in addition to it growing and dividing, which is one of the hallmarks of a cancer cell, it also is dying and shedding parts of those cells. And so while we always think about these cancer cells dividing and growing, an important part of it is that some cancer cells are also dying at the same time. And these shed pieces of these cells or these dying cells can actually be taken up by cells within the immune system. And we call these cells antigen presenting cells. What an antigen is basically a protein that the immune system can see in this situation. And as the name might imply, these specialized cells basically present these proteins to the immune system. And so in a normal context, we might have a cell that might be infected by a virus, as an example. And so in that situation, these cells that are infected by viruses also die and shed proteins. And these viral proteins would get taken up by these antigen presenting cells and basically teach T cells within our immune system to actually recognize these viruses. Well, in the context of cancer, what we think is going on is that these cancer cells may also be shedding these proteins or these antigens, and those may be, in fact, also teaching our immune system, teaching these T cells to actually recognize the cancer cells. And so these T cells could then go back to the sites of tumor and potentially recognize those tumors. Now, an important aspect is when we think about these antigens, we need to think about these as being potential targets that we can go after with regards to immunotherapies. And I'll elaborate on that a little bit. But I really want to introduce this idea that this process may actually be going on within a patient, but there may be mechanisms which actually can turn it off. And so we'll talk about some of the brakes on the immune system, the immunologic checkpoints like CTLA4 and PD-1 that could potentially turn the immune system off and basically render this type of cycle ineffective in the setting of cancer. Now, what we believe happens in people is that we may have a cancer arising from our normal tissues. And over time, these tumors may grow out. And in fact, some of these may be recognized by different components of the immune system. And so I talked about the T cells and the NK cells and other components of the immune system. And what may happen is that these immune cells may actually pick off the cancer cells as they arise. And in fact, some tumors may actually be completely eradicated by our immune system, and we actually may not know that these cancers arise. And where we have seen this actually demonstrated is if you think about settings where patients are immune compromised, such as patients who've undergone organ transplantation and require immune suppression, to prevent their immune systems from rejecting an organ that they might have received. Or if you think about an infection such as HIV that renders the immune system ineffective, you actually can see more tumors growing out in those particular patients. Now, in a normal individual with an intact immune system, what is thought to happen is that the immune system reaches a point that's called equilibrium, or in essence, like a stalemate, where the immune system's sort of picking off tumor cells, but the tumor cells are actually changing and mutating just because we know that these cancer cells have unstable genes in them that basically can allow them to develop mechanisms of escape. And over time, what happens is that these cancer cells that have learned how to escape immune recognition and immune surveillance basically can grow out and develop into bona fide tumors where they've either developed ways to turn off the immune system or they've developed ways to avoid recognition by the immune system. Now, what is some evidence that this type of immune surveillance might happen? Well, as it turns out, in the setting of colorectal cancer, it's actually been shown elegantly by a group in France, Galland et al., that has shown the importance of the presence of T cells within colorectal cancer itself. And so this is just an immunohistochemical stain that's basically labeling T cells within the tumors. And the important part here is what they've shown in different studies is that if colorectal cancer tumors that are at the time of surgery taken out and looked at under the microscope for these T cells, if there are a lot of T cells there, these patients actually do much better than patients who don't have these particular T cells. And that's important in terms of overall survival and their chances of disease progression. Now, if we actually look at where these T cells are localized, whether or not they're actually at the invading margins of the tumors, or whether or not they're in the center of these tumors, or if we look at where these T cells are, the important part is having T cells that get to the tumor, whether they make it into the tumor or to the edges of the tumor. Either of those actually are associated with improved survival if you have a higher number of these. And so these would suggest that these T cells are basically being recruited or going to the sites of tumor and potentially seeing something that's there. And that may ultimately reflect a different biology in the cancer, or it may reflect how the immune system may be recognizing the cancers in these patients. Now, what could these T cells be seeing? Well, T cells recognize these antigens that I introduced that are inside the tumor cell. They get presented, and if a T cell has a receptor that can see that particular antigen or that particular protein, it basically can engage and become activated and can kill that cell. That's how the immune system recognizes cancer cells. That's also how the immune system can recognize virally infected cells. And so what we think is, in the setting of tumors, you could have different proteins that are basically altered within those cancer cells that might distinguish it from a normal colon cell. And so in this case, one example is thinking about mutated self-proteins. And as we talked about with cancer, the genes can become altered or mutated and lead to proteins that are in themselves altered or mutated. And the important part here is the immune system is actually great at recognizing things that are different from ourselves. What they have a more difficult time in recognizing are actually proteins that are in our normal tissues. And so we now believe that these mutated self-proteins that are present within the cancer cells might actually be an important way by which the immune system, these T cells, may be able to recognize colon cancer. Alternatively, we could think about these self-proteins, so proteins that are expressed within normal tissues as well as cancer cells. One of the things we found is that cancer cells may be distinguished by overexpressing or having too high a level of expression of these proteins. And because there's more of these proteins around, you basically will have a tumor cell that has more of these antigens on its surface. And that might be another way that T cells may be able to recognize antigen. And again, in the setting of viruses, some cancers are actually associated with viral infections, and those may actually provide antigens that the T cells may, again, recognize. So now that we have a sense with regards to how immune responses are important for colorectal cancer, I'd like to now talk about cancer vaccines and what has gone on within this field. And so the thinking about cancer vaccines, when we think about immunotherapies, a way we could simplify in terms of thinking about these are immunotherapies that are antigen-targeted immunotherapies versus immunotherapies that we call immunomodulatory therapies. And within this, we think about these immune checkpoint inhibitors that we'll talk about later. But for this portion of the talk, what I'd like to do is discuss these antigen-targeted therapies, which we think about for vaccines. And so this is a slide that I borrowed for a review that we wrote on prostate cancer, but it's also applicable for colorectal cancer. The important point here is we can think about vaccines such as Provenge for prostate cancer or a virus being a way to induce an immune response to specific antigens. And by priming an immune response, we can then have these immune cells target the tumor. What we'll talk about later are these immunomodulatory treatments that require on amplifying a pre-existing immune response. And so within this area of antigen-targeted immunotherapies, we can think about dendritic cell vaccines. We can think about viruses that are basically transduced or modified to express certain antigens. And so I'll introduce in this context a pox virus called PanVac that is being used in colorectal cancer. But there are also other ways, including transduced tumor cells, as well as vaccines that target other antigens that are expressed in cancers. And again, thinking about tumor antigens, these can be tumor-specific antigens or associated antigens, or ones that are derived, what we call oncofetal proteins as potential targets. Now, one approach that I'll first introduce is this notion about a dendritic cell vaccine. And so you could basically introduce an antigen into a dendritic cell, have that dendritic cell go teach a T cell, and to generate T cells that can recognize the cancer cells. And one approach that we had been involved with is a dendritic cell vaccine trial that targeted Carcinoembryonic Antigen, or CEA. This is a protein that's overexpressed on the vast majority of colorectal cancer cells. And basically, the approach that we undertook was to isolate dendritic cells from a patient, load those antigens with Carcinoembryonic Antigen, a peptide derived from that, and then give that back as a cellular vaccine to a patient. And we published on this actually over 10 years ago, where if you give a patient such a vaccine, this is basically just an immune assay to see whether or not we can detect T cells that see this antigen. And so the main point is, if you look in this dotted box, before versus after vaccination, we could actually generate T cells that recognize what we were vaccinating the patient to. And in fact, on this clinical trial, we could actually see regression of metastasis, in this case, lung metastasis within a patient with colorectal cancer. This is a CT scan looking at the lungs being sort of the black areas here, the heart sort of this white thing in the middle, and the main point here is this big white component is a large lung metastasis that basically goes away with this type of dendritic cell vaccine. Now, another vaccine strategy that I'll introduce is PanVac. This is a vaccine that targets CEA, so Carcinoembryonic Antigen that I talked about, as well as MUC1. And it also includes this element that's called TRICOM. These are basically adhesion and co-stimulatory molecules that actually are expressed on antigen-presenting cells. But by including them within this viral vector, any antigen-presenting cell that gets infected will express not only these antigens, but also express more of these molecules that'll make them better antigen-presenting cells. And so with this strategy, you immunize with a vaccinia virus related to the virus that causes smallpox that expresses these elements, and then you boost with another virus that again expresses these elements. And so the point here is, if you think about this virus, it basically goes in and infects cells and basically introduces antigen into these dendritic cells to again prime this T cell immune response. And so the main point I want to raise here is this virus has been actually worked on for over a decade as well, but in a recent publication by Morse et al. Just last year, they actually compared Panvac to dendritic cell vaccines and basically showed in terms of clinical outcome that they were actually pretty similar. And what they also showed was that patients who received this Panvac actually did well compared to a historical control group. This was not a randomized trial, so I don't want to overemphasize this result in that they were comparing to a group that was not randomized to a placebo, but when they compared it to historical controls from their institution, these vaccinated patients appeared to do better following surgical resection. Now, to focus on the final element of immunotherapy in colorectal cancer that I'll touch upon are these immunomodulatory treatments. And so when we think about immunomodulatory treatments, there are actually a whole host of what we now know are important molecules by which T cells can interact with these antigen-presenting cells. And as it turns out, if you generate antibodies that basically target some of these different molecules, you can actually enhance an immune response. And so the first one that I'll introduce or focus on is CTLA-4. And so what CTLA-4 is it's essentially a brake on the immune system. So the T cells, when they interact with antigen-presenting cells, see what we call signal 1 and signal 2. This is a go signal for the T cell to become activated. But once these T cells become activated, they actually now move this molecule CTLA-4 to the surface that basically serves as an immune brake and turns the immune system off. And what's been borne out is that if you actually use an antibody that blocks CTLA-4, this seminal discovery by Jim Allison showed that using this blocking antibody could actually now reverse this brake and actually amplify a cancer immune response. And in fact, this was shown to improve overall survival in the setting of melanoma. Steve Hode's seminal paper in the New England Journal basically showing that patients who received an anti-CTLA-4 antibody or ipilimumab had better survival compared with patients who received a peptide vaccine. And this led to the FDA approval of ipilimumab in the setting of melanoma. Well, what about colorectal cancer? In fact, we participated in a study that looked at Tremelimumab, another anti-CTLA-4 antibody in the setting of colorectal cancer. And what we found was that 1 out of 45 patients treated on this study actually had some response. So again, these are CT scans showing a pelvic mass shrinking, as well as in this case, showing an adrenal mass that shrunk with treatment, but only 1 in 45 patients actually responded. And so based upon this, it was interpreted as not having very much activity in colorectal cancer. But an important aspect of this is when you looked at patients who were alive greater than 6 months, actually almost half of the patients were alive at this time point. And an important aspect is with anecdotal patients from this trial, including patients at UCSF, we've actually seen patients years after participation in this study who, based upon the statistics, we would have thought would have unfortunately died from the cancer. An important aspect of this is that the majority of these patients, in fact, all of these patients, except for this one patient, received multiple doses of this antibody because the other patients were taken off either because of side effects or because they had disease progression as we measured them with conventional means. But we believe that some of these patients may have actually gone on to have clinical responses. Now, switching gear to the final immunotherapy that I'll highlight is PD-1, and there's been a lot of excitement around antibodies targeting PD-1 and PD-L1. This immunotherapy, rather than working on T cells as they become activated with anti-CTLA-4 antibody, are thought to be important in terms of rescuing T cells that are now interacting with tumor cells. These activated T cells express PD-1. Tumor cells can actually express the ligand for these and turn the T cells off. And so the thinking here is that if you use an antibody that blocks this interaction, you may actually rescue these T cells that are sitting within the tumor site. And in fact, if you look at PD-L1 expression, that actually can be associated with a worse prognosis in a lot of different cancers, including GI malignancies. And so these are just examples of these malignancies where if PD-L1 is expressed on those tumors, those patients unfortunately do worse with those tumors. Now, if we actually look at colorectal cancer, actually a significant proportion, more than half of colorectal cancers actually express PD-L1. And so this is, again, a molecule that could turn off the T cells that ultimately make it there into the tumors. And in fact, if you look at expression within tumors that express PD-L1, you can actually see T cells within that tissue compartment, again, arguing that T cells that make it there may actually have a PD-L1 there that might ultimately turn them off. And so again, seminal studies that were published now a couple of years ago in the New England Journal basically showed broad clinical activity with both antibodies targeting PD-1 as well as antibodies targeting PD-L1. And the important aspect of this was, in addition to melanoma, this actually showed activity in other cancers that were unexpected, one of which was lung cancer. And so there are now large phase 3 clinical trials in a lot of different cancers, including lung cancer. But with these initial reports, there was actually no reported activity in colorectal cancer. This is just showing the clinical activity in melanoma and kidney cancer and lung cancer. And the main point of it is these are what are called spider plots, which basically track the disease burden within patients. If these lines go down, that means that the amount of tumor within the patients goes down over time. But an important aspect of this is you can actually see patients that actually have progression of their disease followed by then regression of their disease. And as I alluded to with the anti-CTLA-4 trial in colorectal cancer, a lot of our trials, once a patient has any evidence of progression, they get taken off of these studies. But what we now know to happen with immunotherapies is that we may see this pattern of cancer getting worse before it gets better, so that our newer trials basically allow for this interval progression before taking a patient off a clinical trial. And so these are, again, showing responses to these patients. This is showing that the majority of patients on these trials actually had responses. But what about colorectal cancer? There was no responses to colorectal cancer. But in a recent update published last year, in fact, there was a clinical response with regards to colorectal cancer. And this turned out to be a durable response. And so this is basically an involved lymph node of a colorectal cancer patient that actually responded. And the important point here is you can see that this patient's tumor was ringed by PD-L1, the molecule that could turn off an immune response. And you can see that this tumor is also ringed by immune cells, CD8 T cells, that basically were there at the tumor surface. And so this would actually indicate that there is clinical activity with anti-PD-1 antibodies. And in fact, with the anti-PD-L1 antibodies, there was actually reported clinical activity at the last ASCO meeting in a participant on that trial. And so I think, an important aspect is there appears to be clinical activity with these immunomodulatory treatments as well. And so with that, what I wanted to do was actually conclude and then just touch briefly upon the challenges that we face in the field. So with regards to conclusions, I hope I've shown to you that the immune system can actually see colorectal cancer, and that recognition is actually important in that it actually is associated with improved survival in patients. We now have antibodies that target molecules that are either shed by colon cancer or actually expressed on colorectal cancer, and these are FDA-approved treatments. Cancer vaccine development has continued, and this has shown preliminary evidence of activity against this disease. We also have evidence of activity with these immune checkpoint inhibitors, but a key area is we really need to improve the activity of immunotherapy in colorectal cancer to really make an impact. Points to consider: what are the settings that may be most relevant for treatment? And because we have other treatments for colon cancer, treating patients who've actually gone through all these other treatments may be potentially too late, and their immune systems may be too beat up to potentially respond to these immunotherapies. And so we need to think about that. In terms of clinical endpoints, the important part with immunotherapy is we've seen improvements in overall survival without improving progression-free survival. We also see these delayed responses, and so we need to think about these when we design trials. Well, we can't use conventional response criteria as we've used in the past. We also need to develop biomarkers, ways that we can identify who's going to respond to a treatment and who's not going to respond to a treatment. And another aspect that I think is incredibly important is thinking about combination therapies or sequencing of therapies. In cancer, we do not think about using a single drug by itself. In fact, with all of our current therapies, they actually don't work very well as single drugs. And why should we expect any different from our immunotherapies? And so I think this really gives us an opportunity to think about how we can incorporate immunotherapies with conventional therapies, as well as anti-angiogenic therapies, other targeted therapies, and another aspect in terms of combining immunotherapies. And with regards to combining immunotherapies, Jed Walchuk published in the New England Journal a combination of an anti-PD-1 and an anti-CTLA-4 antibodies targeting PD-1 and one targeting CTLA-4. In this combination trial, putting the two together was actually better than using the drugs sequentially or individually. And so this really gives us hope that combining these different immunotherapies may also further enhance the activity of immunotherapies. And so with that, I'll close and move on to our questions.
All right, well, thank you very much for that, Dr. Fong. Lots of questions from the viewers. Some of them you actually answered throughout the presentation, so you get points for that. So let's kind of work backwards here. You just discussed combination therapies, and as you said, there's a lot of excitement about the potential of these combinations of drugs to work together to create a stronger and more effective immune response. And we've seen that combining anti-CTLA-4 and anti-PD-1 in melanoma does wonders, as you showed us with that very beautiful chart from Dr. Walchuk. But the question is specifically, have you seen similar effects in colorectal cancer, either combining active immunotherapies or any of the other drugs or therapeutic strategies that you talked about?
We have not seen that at this point because we actually don't have any results from these clinical trials. That said, because there are multiple companies that have anti-PD-1 antibodies as well as anti-CTLA-4 antibodies, those combination trials are actually forthcoming. The initial experience that Dr. Walchuk published was in the setting of melanoma, but there are now multiple trials in different diseases. And so colorectal cancer is one of the diseases that there's interest in, but we don't have any results at the present time.
Okay, thank you. Another thing that you mentioned was in your consideration, your list of considerations, is the time at which an immunotherapy is given to a patient and the types of treatments that they've received prior to this, and how that could potentially have a negative impact on the vitality of the patient's immune system and its ability to respond to immunotherapy. So has there been any movement in bringing these to earlier stage cancer patients?
Well, there's a lot of interest in doing that. One of the challenges that we face now is one of resources and access to these different drugs, and we're working with the different companies to develop these treatments in patients with earlier disease. One of the areas that we're working on is actually giving these immune-based treatments prior to surgical resection. That would be a setting where the patients hadn't received prior chemotherapy to really understand how these treatments might work within the tumors. But those are really small trials that are focused on trying to understand how these treatments are. I think we're continuing to work with the different pharmaceutical companies that have these drugs to really study these in patients with earlier disease rather than focusing on patients who've unfortunately progressed following different lines of chemotherapy. And so that's something that we've continued to advocate and push for, and we look forward to developing those trials in the future.
Okay, thank you. And this is a question that was asked. Not everyone understands the difference between a phase 1, phase 2, or phase 3 clinical trial, and you mentioned that these checkpoint blockades are in large-scale trials. Can you just place that within context from the time of discovery to the time of FDA approval and when patients can get access to these drugs on the market?
Well, the great thing now is how that timeline has really accelerated. So if you look at CTLA-4, anti-CTLA-4 antibodies, the initial mouse experiments were over a decade ago, probably on the order of about 15 years ago. And that took quite a bit of time for that drug to actually be developed so that it was FDA approved now 2-3 years ago. And part of that was there was a healthy skepticism in the oncologic field in terms of whether or not immunotherapies actually worked. I think in 2014, we no longer need to convince companies and granting agencies that immunotherapy works, and that's really accelerated the timeline. And so from the phase 1 trials of anti-PD-1 antibodies now to the phase 3 clinical trials, that timeline has really been compressed to on the order of 2 to 3 years. And in fact, many of the phase 3 trials with anti-PD-1 antibodies have finished accrual. In the setting of melanoma, we've been involved with a kidney cancer trial. And so those results should be available over the next year or two, where we're hoping either at the end of this year or sometime next year, we will have FDA-approved anti-PD-1 or anti-PD-L1 antibodies. That unfortunately will not be in colorectal cancer, but what we would be looking for is as these companies continue to broaden out their experience in other diseases, including colorectal cancer, that it will be subsequently studied in those diseases.
Okay. Something that I don't think you specifically addressed in your talk today, but has been discussed in different tumor types by presenters in previous webinars, is the subject of adoptive T-cell therapy and chimeric antigen receptor T-cell therapy. Now, these CAR T-cells have generated a lot of excitement. They've made major headlines because of their ability to completely eradicate some forms of leukemia. So the question is there any movement in applying those technologies to colorectal cancer?
There absolutely is interest in doing that. I think the CAR technology is a really exciting technology. One of the challenges with colorectal cancer is that unfortunately we don't have an antigen, that immune target that's necessary for these CAR T cells like we do in leukemia. The results targeting CD19 are great in leukemia, but that target is not relevant for colorectal cancer. And people have tried targeting other antigens, including CEA, and unfortunately those have led to not just some responses within the tumor, but actually development of colitis because CEA is also expressed on the colon. And so I think people within the field have really been now revisiting what are the antigens that are that we could potentially use in the setting of CAR therapy for colorectal cancer, and that work is all ongoing.
Has there been any work in developing immunotherapies that target lipids or carbohydrate-based markers of colorectal cancer?
Well, that's an area that some people are interested in and are studying. I think these molecules are also very interesting in terms of how the immune system could potentially recognize antigens. One of the antigens that I briefly touched upon is MUC1, which can be aberrantly glycosylated in the setting of malignancy. And I think there are all these different ways that the immune system can focus on them, and that could represent another way that the immune system could recognize colorectal cancer.
Okay. And so you mentioned colitis as one of the potential side effects, and we've heard about these autoimmune reactions that can occur when patients receive these checkpoint blockade. And so the question is, what is the way to deal with that? What does a patient do, or what does a doctor do when his or her patient is beginning to show symptoms of the beginning of an autoimmune response after receiving one of these treatments?
Well, this is one of the important points now, that as we develop greater experience with dealing with some of these side effects, we now have treatment approaches where, for instance, with colitis, if we have any signs of a patient developing diarrhea, we actually have very specific medications that we use to actually address those treatments. And so while patients may develop diarrhea, we now have approaches to really control that diarrhea so that doesn't represent a potentially life-threatening side effect. With the Tremelimumab study in colorectal cancer, we did see a fair number of those patients develop diarrhea, and at the time, that experience wasn't as significant as it is now. Now that, for instance, ipilimumab is FDA approved, by and large, all of our oncologists are now very comfortable with dealing with those side effects. And I think those will be very manageable as these treatments come forward.
All right. Well, unfortunately, that's all the time that we have for today's webinar. We had many interesting questions, which I'll do my best to forward on to you, Dr. Fong, in case you have some questions, particularly from clearly some of their participants are scientists, or they're very well-educated patients about this subject. So we'll try to pass on those more complex questions to you for possible response. In the meantime, I'd like to encourage everyone to go to cancerresearch.org/webinars to stay up to date on our next offerings. We do plan to have one next month. That date has not yet been set, unfortunately, but you can find out by going to cancerresearch.org/webinars. You can also access all of our previous webinars, which we began last year, so there's a good number of them there. And I'd also like to point out that this June is our second annual Cancer Immunotherapy Month, and we'll be offering a very rich calendar of events throughout that month, so please look forward to receiving some information about that, and you can join the movement. So you can find us on Facebook, on Twitter, and of course our blog, cancerresearch.org/blog. I'd like to thank all of our participants for joining us today, and Dr. Fong, I would especially like to thank you for this wonderful presentation, and I wish you all a very